Vaccine. What is vaccine? A vaccine is a biological preparation that improves immunity to a particular disease. A vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of the microbe. The agent stimulates the body's immune system to recognize the agent as foreign, destroy it, and keep a record of it, so that the immune system can more easily recognize and destroy any of these microorganisms. Types of vaccine: live attenuated vaccines, inactivated vaccines, subunit vaccines, toxoid vaccines, conjugate vaccines, DNA vaccines, and recombinant vector vaccines. First, live attenuated vaccines. Live attenuated vaccines contain a version of the living microbe that has been weakened in the lab, so it can't cause disease. Because a live attenuated vaccine is the closest thing to natural infections, these vaccines are good teachers of the immune system. Examples: vaccines against measles, mumps, and chickenpox. Inactivated vaccines. Scientists produce inactivated vaccines by killing the disease-causing microbe with chemicals, heat, or radiation. Such vaccines are more stable and safer than live vaccines because dead microbes can't mutate back to their disease-causing state. Examples: vaccines against influenza, polio, hepatitis A, and rabies. Subunit vaccines. Instead of the entire microbe, subunit vaccines include only the antigens that best stimulate the immune system. In some cases, these vaccines use epitopes, the very specific parts of the antigen that antibodies or T cells recognize and bind to. Because subunit vaccines contain only the essential antigens and not all the other molecules that make up the microbe. Examples: hepatitis B and A cellular pertussis. Toxoid vaccine. For bacteria that secrete toxins or harmful chemicals, a toxin vaccine might be the answer. These vaccines are used when a bacterial toxin is the main cause of illness. Scientists have found that they can inactive toxins by treating them with formalin. Such detoxified toxins called toxoids are safe for use in vaccine. Example: tetanus toxoid and diphtheria toxoid. Conjugate vaccine is a type of vaccine which combines a weak antigen with a strong antigen as a carrier so that the immune system has a stronger response to a weak antigen. Most commonly, the weak antigen is a polysaccharide that is attached to a strong protein antigen. Examples: Haemophilus influenza type B vaccine. DNA vaccines. Still in the experimental stages, these vaccines show great promise and several types are being tested in humans. DNA vaccines take immunization to a new technological level. These vaccines dispense with both the whole organism and its part and get right down to essentials, the microbe's genetic material. Example: influenza vaccine. Recombinant vector vaccines. Recombinant vector vaccines are experimental vaccines similar to DNA vaccines, but they use an attenuated virus or bacterium to introduce microbial DNA to cells of the body. Vector refers to the virus or bacterium used as the carrier. Example: DPT. Difference between vaccination and immunization. Vaccination is the administration of an antigen material to stimulate an individual's immune system to develop adaptive immunity to a pathogen. Immunization is the process whereby a person is made immunoresistant to an infectious disease, typically by the administration of a vaccine.